Hello everyone. So today we are going to continue our lecture about the biochemistry from USMLE Step-1 book. So in the previous lecture we already discussed about the chromatin structure in detail. So now let's review it. So the chromatin structure we already discussed. What is a chromatin structure? It is made up of 15 to 20 million 15 to 20 million nucleosomes. Okay, so chromatin is made up of 10 to 15 million of nucleosome. So what was this nucleosome again? Let's get here. Nucleosome is composed of a DNA, histone protein, and histone 1 protein. Okay, so a nucleosome is made up of these three things. Like how this is a histone protein, and the DNA is wrapped up twice around this, and it is going like this and again like this, looping twice around these histone proteins. And where is H1? H1 is the linker protein which is linking all these nucleosomes together. Okay, linking these uh, uh, histone and DNA complex together. Okay, so this whole is called, or you can say these groups. I have made just three of them, but there is usually 10 to 15 million of nucleosome in one chromatin. This whole is called a chromatin. Okay, so now let's go into a little bit detail of this. Okay, so now let's see where is a chromatin actually is. Where is chromatin located? Let's see where it actually is located. So let me make this is a circle. What is this? This is a cell. Okay, and what is inside here? There is a nucleus. Okay, and what is inside this nucleus? This is our chromatin. And what else is there in nucleus? There is nuclear pores here, like this. These are called nuclear pores. This whole is nucleus, and this is our chromatin here. And inside, what is the nucleus? Chromatin, nuclear pore, and this whole is a nucleus. And nucleus is inside this big thing. What is this big thing? This is cell. What is these around here? This is a cell membrane. And what else will be inside the cell? There might be this mitochondria, and there can be ribosomes, and there can be Golgi bodies. So I'm just making a nutshell. What is a cell? Everyone knows it is the basic structural unit of life. From a cell, everything about our body starts. Everything begins like this. Cell will divide and it will replicate, and there will be our skin will be made, or liver will be made like this. with the help of cell, all the complete body of a human being is formed. But how these cells are replicating and how these cells get these informations to make these Golgi bodies, ribosomes, mitochondria, and everything, how they are making these proteins for cell membrane, phospholipids, how they are doing it? They are doing it with the help of this chromatin. So now let's move on to our next step in our USMLE biochemistry book. This is our book here. And what is the next topic here? You can see it is heterochromatin and euchromatin. After chromatin structure, which we discussed in the previous video, our next topic is heterochromatin and euchromatin, DNA methylation, histone methylation, histone acetylation, and histone deacetylation. Now we are going to cover these topics and understand it correctly. When you look at these topics from this book, you, after studying the chromatin structure, it straight away says heterochromatin condensed, appear darker on electron microscope, sterically inaccessible, transcriptionally inactive, increase methylation, decrease acetylation. But you just can't understand what is heterochromatin and what is the relation of heterochromatin with chromatin, or are there three types of chromatin? One is normal chromatin, what is heterochromatin, what is and other is euchromatin, and there is other thing like the DNA methylation, because they are not explaining it very well. So if you guys want to understand this, then you have to go a little bit deeper, and you must you definitely need a tutor who will teach you. So I'm going to teach you what is this. So now let's look at this. We just discussed that chromatin is the most important part from where these cells and the things inside the cell will originate, and from this chromatin more cells will come and a body will be formed. Why? What is important in chromatin? What is the most important thing? It is DNA. Okay, DNA is the most important thing, because of which all these things are forming. Okay, so now let's discuss heterochromatin and euchromatin. What are those things? So now let's look at this. We already know what a chromatin is. Now let's see. Heterochromatin is actually a part of chromatin, or you can say is a structural differentiation of chromatin. Like one is heterochromatin, other is euchromatin, and there is a third part which was not mentioned in the book, and that is chromosome. Everyone is familiar with what is a chromosome. Okay, so now let's go into detail. Now see, uh, a heterochromatin has this type of structure. Okay, it is very tight and very tightly packed. And this is euchromatin. Euchromatin is very loose. And this is chromosome. It is hugely tightly packed, but it has two divisible bodies which we can see, so it means it can easily divide. So first thing I will clear it early. Chromosome is that shape of chromatin. These three are the shapes of chromatins. Okay, first remember this. When the chromatin is very tightly packed, it is called heterochromatin, and it looks like this. When chromatin is loosely packed, then it is called euchromatin. They are just giving it names. When it is loosely packed, they gave it a name, euchromatin. When it is very tightly highly condensed, then it is called heterochromatin. and when it is extremely compressed during the cell division time, then it gives this type of shape. So they named it chromosome. Okay, it is, this shape is seen when there is cell division. During cell division, chromatin have this shape, the shape of chromosome. Okay, so I'm just discussing this part separately so that we can easily focus on those two topics. Okay, so now let's go into the detail of heterochromatin. Okay, first thing, I already told you about the heterochromatin. One thing that we can understand from its name, from H as well, that it is highly condensed or tightly packed. Now what does this mean? It means that when a heterochromatin is tightly packed, then it is highly condensed. And what will happen if it is tightly packed? We must know that what DNA is doing before that. Okay, what is the importance of its tightly packaging and loose packaging? Like, look, when this DNA has to convert to RNA, and then RNA has to form structural or functional proteins. So what is the structural and functional proteins? Liver has a protein, a group of cell is called protein. Okay, so when RNA makes a lot of protein, then it means it has made a lot of cells, and they combine and make a protein, and a group of protein makes an organ. So liver will be formed if there are a lot of proteins. So RNA is going to make structural and functional proteins, like what kind of structure any organ has to have, and what kind of function any organ has to have. So according to that, organs will be formed. So RNA is a must for that. But the most important thing will be DNA. From DNA, the master plan will come. Like, if a monkey is a monkey, why he is a monkey? Because he has a DNA of a monkey. If an elephant is an elephant, why he has a huge trunk? Because he has a DNA of that huge trunk. DNA will be transcribed to RNA, or will be made into RNA in simple words, and RNA will be made into proteins, and this protein will help in the structure of that trunk, so that elephant will have that trunk. Okay, so uh, the most important thing is that DNA has to be converted into RNA, so that all these things will form. And the conversion of DNA into RNA is called transcription. Okay, so transcription is a must for any organism to form completely, or any organ. If there is any organ which has to form, it has to form after transcription. So what is going on in here in heterochromatin? Heterochromatin is tightly packed. Okay, so you know how this transcription actually occurs. The DNA is, when the DNA is loose like this, only that time enzymes can come and act on this DNA, and then only then it can uh, make the RNA. But if the DNA is very tightly packed, if DNA is like this, very tight, then the enzymes cannot act on it, and transcription will not occur. So what is the second step here? Second characteristic of heterochromatin, first thing is that it is highly condensed. Okay, so if it is highly condensed, what will automatically happen? It will become transcriptionally inactive. Okay, so this was the thing which was mentioned in the book of USMLE as well, that heterochromatin is highly condensed and is transcriptionally inactive. Now you guys know very clearly what does this mean. It is highly condensed means it is highly tight, so that enzymes cannot work on it and cannot do transcription. No transcription, no RNA, and then thus this DNA is inactive, or this chromatin is inactive. It is not divisible right now, it is not working. You can say it is a mute DNA here. So that is exactly what is written in your USMLE book if you are studying from that. So what is the third characteristics? If it is very condensed, if something is very condensed, look, this one I made loose and this one highly condensed. Now which one is looking more darker, this one or this one? Which one is looking more darker? Definitely this one. Why? Because it is highly condensed. So a highly condensed heterochromatin will appear darker on electron microscope. This is the third characteristics of heterochromatin. And what will be next? There, and we before and going to the fourth point, we need to understand one important thing as well. Why it is highly condensed? What is the thing which is making it highly condensed? Okay, we must understand that first. Now just look at this. Why something is highly condensed? Why these DNA is becoming highly condensed? Look, this is a DNA. Usually a DNA had, everyone knows, adenine, guanine, cytosine, thymine. These four are the nitrogenous bases. We will talk about it in detail in the lecture of DNA structure. But when something will come here, like for example, a problem will come. I'm making this problem CH3. Okay, here comes CH3, here comes CH3, here comes CH3, here comes CH3. The DNA was pretty loose before this, but what happened? These groups started coming here and attaching to this cytosine and thiamine. I just made it here, but it is not actually attaching with adenine and guanine. A group came here and it attached with cytosine and thiamine. What happened? DNA became condensed. This is how it is becoming condensed, and what this process is called DNA methylation. This was the topic, this was the heading there in USMLE book about DNA methylation. So what is the thing which is actually making that heterochromatin or that chromatin condensed? These methyl groups, this CH3, this is a methyl group. These methyl groups are the one which are responsible for the condensation of the heterochromatin. So what will happen? This is a DNA. There is adenine, guanine, cytosine, thiamine, another adenine, guanine, cytosine, thiamine, like this. So this methyl group will come here, attach here with cytosine and thiamine, and it will start making it condensed. And there is another thing as well. There are also nucleoside in between, histones in between here. So what histones have here? They have lysine and arginine groups, L and A, lysine and arginine groups. So these methyl groups also start attaching with these two as well. Like, let me make it here again. This is DNA and this is histones. This is adenine, guanine, cytosine, thymine, adenine, guanine, cytosine, thymine, and there are more histones here and here. And what does histones have? Lysine and arginine group, lysine and arginine group, lysine and arginine. So what will happen? Methyl group will come attached with this arginine and lysine group, and here as well, and with cytosine as well, and again with lysine and arginine, and again with cytosine and thymine. So now look how condensed this is starting to become. Actually attached with guanine, not thymine, it was a mistake, sorry. So it is attaching the cytosine and guanine, and we can say that there are two types of methylation are occurring here: CpG methylation and La methylation. Like these two groups are getting methylated here, methyl is coming here, and what is it is doing here? It is definitely making it hugely condensed. So when it is becoming hugely condensed, what will happen? No enzyme can come here and work here to make RNA. No RNA will be made because no enzyme can work here and make RNA. It will be not possible. So this DNA or this chromatin will become transcriptionally inactive, and it will be called heterochromatin. So what about that other chromatin? Let's go here. There is other type of chromatin which is called euchromatin. Okay, so what are the characteristics of euchromatin? It is loosely packed. Definitely this is the difference between heterochromatin and euchromatin. Euchromatin is loosely packed, heterochromatin is tightly packed. Okay, so if it is loosely packed, we definitely know what will happen. It will become transcriptionally active, and if it is transcriptionally active, it is loosely packed. What will be the other thing that will happen? It will appear lighter on the electron microscope. Like I showed you, the condensed one is darker and the loose one is lighter, so it will appear lighter on electron microscope. So we know why heterochromatin is tightly packed, because of methylation, DNA methylation and histone methylation. When these methylation will occur, if only one, if only DNA is methylated, then still it will be tight, and if histone is also methylated, then still it will be uh, condensed, and if both are methylated, then it will still be condensed. Okay, so transcription will not occur, and it will appear darker on electron microscope. But why euchromatin is loosely packed? What is the reason of its loose packaging? So now let's discuss that too. These two were uh, these were condensed because of what? Methyl group attaching with histone, where on histone? On lysine and arginine group, and on the DNA where cytosine and guanine group. So this is called CpG methylation, or what is its other name? DNA methylation, and this can call La methylation or histone methylation. So these are two types of methylation which are occurring, but what is making this chromatin loosely packed? And that is his histone acetylation. And other thing, histone deacetylation. So now what are these two things? Look, we already discussed in the previous lecture that when DNA and these two, why are they both are packed? This histone and DNA, why they are both packed? Because DNA has negative charge. DNA has a negative charge and histone has positive charge. So what will happen? An acyl group will attach with it and it will do acylation. So just remember, is acyl group came and attached with histone, and histone acetylation will occur. And what will happen? Positive charge will be gone. Positive charge will be gone, and what will happen? The attachment between these two, histone and DNA, will become very loose, like this. It will be very loose, and what will happen? The enzymes will come and act on it, and it will make RNA, and thus what will happen? It will be transcriptionally active. So this part is very easy. And histone deacylation is just another topic which will make it harder again, or condensed again. Deacylation means positive charge will come back on histone and it will be condensed, but this is not related with it, but this is another term which was used in the book. But main thing is histone acetylation is occurring, which is making it loosely packed. So now what we know now, let's look at it again. The characteristics of histones. Characteristic of heterochromatin and euchromatin. What is heterochromatin? It will be tightly packed. There is no doubt about it now. It will be tightly packed, and euchromatin will be loosely packed. Why it is tightly packed? Because of DNA methylation. So what will be happening here? Increase DNA methylation and increase histone methylation. And what will be happening here? Increased acetylation. And what other things will be happening here? It will appear darker on electron microscope. It will appear lighter on electron microscope. And what is the most important thing? This will become transcriptionally inactive, and this will be transcriptionally active. So everyone must have understood this by now. Heterochromatin tightly packed. Why it is tightly packed? Because of methylation. It appears darker on electron microscope, and because of its tight packaging, it is transcriptionally inactive. What about euchromatin? It is loosely packed because of acetylation, so it will be transcriptionally active. So these are the two types of chromatin based on their structure, and the third type was chromosome, which is only seen during cell division. So everyone must have understood it. Now let's look at the book again. What it says, it says heterochromatin is condensed. We know why it is condensed, because of methylation. It appears darker on electron microscope, sterically inaccessible, which means it is tight and it is not accessible for transcription. Thus it is not sterically accessible, so it will be transcriptionally inactive. There will definitely be increased methylation and decreased acylation. Now you guys must have understood what this how difficult it looked before opening up the lecture, or uh, opening up this topic. And heterochromatin highly condensed, no doubt about it now. And bar bodies are the things which are visible on the periphery of the nucleus, because these are inactive chromosomes. Now, euchromatin, less condensed, it appears lighter on electron microscope. Transcriptionally, it is active, and sterically, it is also accessible. Euchromatin is truly transcribed. Euchromatin is expressed. So everything expressed means it is easy to transcribe or sterically accessible. Both are same terms. And DNA methylation reversibly changes the expression of DNA segment without changing its sequence, and it is involved in aging, carcinogenesis, epigenetics. These are the uh, uh, times when DNA methylation is occurring, and it is stopping the DNA replication and DNA transcription and formation of RNA. And this I told you, CG methylation makes DNA mute by on cytosine and guanine by attaching it to cytosine and guanine. And this is histone methylation. Transcription suppression happens here, and you can remember this from this. Histone methylation mostly makes DNA mute, and methylation makes DNA mute. Okay. And acetylation is removal of positive charge, relaxes DNA, increases transcription, and removal of acetyl group tightens DNA and decreases transcription. So there is no doubt about this now. It is very easy. You just have to understand it first, then it appears very easy. If you guys still have any question about this topic, if there is anything which you didn't understand, because I understand it very clearly, and if you were not able to understand it from my this explanation, then I can still explain it again, and in more easy words as well. So you can just comment below which part you didn't understand, and I will make a video again and in more simpler words. Okay. So this was our topic of complete topic of chromatin structure and its types and its transcriptional activity. Thanks for watching everyone. If you like the video, kindly subscribe the channel and click the bell icon. And if you want lectures on any other type of topic, then you can comment below. I will make a lecture on it. Thank you so much.